Thank you very much for the invitation to present in this scientific summit. So today I would like to present some recent work we have been doing on the mesenchymal transformation of pediatric ependymal tumors. And this is a project on which we have greatly benefit from the resources from CBTN, both at the level of tissue and at the level of data also. Just say this is work that recently was published and is work led by Rachel Ovin, who is sitting there. So Rachel is presenting a poster this evening. And so if you are interested on hearing more about what I will tell you, please stop by. She will be also next year graduating. She will be in the job market, so it will be a good opportunity to talk to her. All right, so as you well know, gliomas are not epithelial cancers. They lack many of the features of epithelial cancers because they are derived from the neuroepithelium. They lack expression of E-cadherin and other markers. However, they can still experience a similar epithelial to mesenchymal-like transformation as epithelial cancers, which is characterized by the expression of mesenchymal markers and hypoxic, angiogenic, glycolytic, and ferroptotic programs. So its signature was first described in the context of glioblastoma, was associated with poor survival, resistant to therapy, tumor inflammation, vascularization, and necrosis. And with the realm of single-cell approaches, it has become evident in the context of glioblastoma that basically every glioblastoma is very heterogeneous, and at least a subset of the cells will present this mesenchymal phenotype. And it has become also evident that not only glioblastoma has this mesenchymal phenotype, but other gliomas can have also this type of phenotype. In our lab, we have been focusing on pediatric ependymoma. I don't need to introduce this tumor to this audience, but just to say that it remains a significant therapeutic challenge, especially in pediatric cases, where it represents nearly 1/3 of the central nervous system neoplasm, and the 5-year overall survival is pretty poor. And part of it is because of the lack of understanding about the molecular pathways that underlie tumor genesis and progression of these tumors. And in part, as it was mentioned in some of the previous talks, this is due to the absence of known mutations in driver or mutations in known driver oncogenes. So 80% of the cases in these populations approximately lack a mutation in some known driver oncogene. So when I started my lab at Penn almost 5 years ago, we decided it was a great opportunity to start investigating more into these tumors for two reasons. One was the availability of resources like CBTN, which have been archiving tissue from these tumors and generating data from these tumors for already quite a few years. And also because of the emergence of single cell approaches that enable to perform or to profile flash frozen tissues by using single nuclei, RNA-seq and ATAC-seq. So we have been working with a cohort of 46 pediatric ependymal tumors in the posterior fossa, for which 42 of them, or most of them, were already profiled by the CBTN at the bulk level for RNA. And we decided to profile 9 of these tumors with single nuclei RNA-seq and single nuclei ATAC-seq, comprising both primaries and distal metastases. I will not go into the details of how this experiment was designed, but I will mention that one of the things that we had to do in order to design and decide which samples to use was to develop a classifier to assign molecular subgroups, molecular subtypes to the different samples based on the bulk RNA sequencing data. So I will, again, not go into the details of exactly how we did and so on, but I will just summarize some of the main findings that we obtained. And one of the first things that came out from the single nuclei RNA sequencing data was that the neoplastic cells in these tumors, as in other tumors, as it has been also noted in these tumors simultaneously by other groups, recapitulate neurodevelopmental radial glia cell differentiation trajectories. There's a lot of heterogeneity within the tumor cells, some of the tumor cells, they express programs that resemble those of more differentiated cells like ependymal cells, astroblasts, neuronal progenitor cells, and a population of mesenchymal-like cells that in our case was mostly enriched in the distal metastases. And apart from this, then there are some set of more undifferentiated tumor cells that have a phenotype more like tumor stem cells. So this finding was important because it enabled us to identify the subpopulation that has a more stem cell-like phenotype, and now we can characterize molecularly. But we were particularly interested on this transition from the undifferentiated cells, which have a more neuroepithelial phenotype, into the mesenchymal-like cells that were enriched in the distal metastases. So we perform standard analysis on this type of data, differential gene expression analysis, pathway enrichment analysis, expression of ligands and receptors. And we found that the data was consistent with tumor cells and infiltrating microglia expressing pro-inflammatory cues. So the tumor cells were expressing genes coding for chemokines that act on receptors in the microglia. And the microglia were expressing inflammatory cytokines which act on receptors on the tumor cells. So we came out with the hypothesis that maybe the tumor cells were proactively attracting microglia into the tumor, and microglia were secreting these inflammatory cytokines, and they were triggering reactive gliosis programs in the tumor cells that then acquire a more mesenchymal phenotype. It was just based on, consistent with this, we look at the tumor cells and perform pathway enrichment analysis on those, and we saw that they have activations of pathways that were compatible with those, with the effect of those cytokines secreted by microglia. So a lot of NF-kappa B signaling, TNF-alpha signaling via NF-kappa B, mTORC, and so on and so forth. So all this was just based on the patients that we, or the samples that we profile at the single cell level. So then we went to the larger cohort of tumors that had been profiled at the bulk level, and using gene expression deconvolution methods, tried to see whether that data were also consistent with this hypothesis. So here we have the 42 tumors that were profiled at the bulk level, arranged from left to right according to the inferred abundance of mesenchymal-like cells. And our findings were again consistent with this hypothesis. So tumors with a higher abundance of mesenchymal cells, we were having also a higher abundance of microglia, higher abundance of vascularization and enrichment for those inflammatory pathways that we were seeing in the single cell data. We then turned out into the single nucleotaxic data, and we observe again a consistent picture, same type of heterogeneity, same cellular processes happening inside the tumor. And we use this data to characterize the enhancers and transcription factors that were associated with that transition from neuroepithelial tumor cells into mesenchymal tumor cells. And we found that the neuroepithelial tumor cells, not surprisingly, were expressing a set of transcription factors that are expressed usually during normal development in the hindbrain and the isthmic organizer, where these tumors arise, and in particular in the neuroepithelium of these regions. And these transcription factors were switched off during the mesenchymal transformation, and instead a set of mesenchymal transcription factors and inflammatory transcription factors like NF-kappa B complex, AP1, MACK, MYC, TAT, were turning on during the transition. I will not go into details, but we look also in more detail at that mesenchymal population, and we found that there is a lot of heterogeneity within the mesenchymal cells. So we can identify multiple subpopulations, and we think they are probably related to different pathways for this mesenchymal transformation, and also related to different combinations of cytokines in the local microenvironment of the cells. So then we try to validate more mechanistically some of these findings, and consider patient-derived cell model from recurrence of one patient, and treated in vitro those cells with some of the cytokines that we were seeing expressed in the infiltrating microglia to see whether we were able to induce the mesenchymal transformation in vitro. And indeed, we found, based at least on the expression of mesenchymal markers that we were seeing in vivo, that we were able to induce that transformation in vitro using some of these cytokines, and also based on a cell migration assay. So just summarizing, so ependymal tumor cells recapitulate cell differentiation lineages from radial glia and can transition into a mesenchymal-like state. So this mesenchymal transformation, we think, is associated with pro-inflammatory signaling from the tumor cells and infiltrating microglia, and involves the inactivation of neuroepithelial transcription factors and the activation of NF-kappa B, AP1, and TAT pathways. Modulation of these pathways with different cytokines lead to substantial heterogeneity within the resulting mesenchymal cells. And I think one of the indications that come out from our study is that given how different the neuroepithelial tumor cells and the mesenchymal tumor cells are in these tumors, prospective therapies to be efficient for this cancer will need to simultaneously target both proliferative neuroepithelial and mesenchymal-like tumor cell populations. I will just finalize acknowledging people who did this work in the lab, especially Rachel and Emma, our collaborators at CHOP and Penn, Maria Rita Santi, who is in this meeting also, and McLean Nasrallah, and of course CBTN and D3V for all these fantastic resources without this study wouldn't have been possible. Thank you very much.